molecule called hemoxygenase 1, HO-1, is a driver or is essential for establishing insulin resistance and side effects associated with obesity in humans and in our model system, the mouse. When we take out HO-1 in hepatocytes or in macrophages, we can either attenuate or completely block the consequences of severe obesity in mice.
We find that HO-1 not only is a driver, but it seems to be a driver that's already acting at extremely early stages of disease. And that's a key, because for diagnosis, you want to diagnose as early as possible. And for treatment, of course, you want to treat as early as possible.
The biopsies we obtained were from metabolically important tissues, namely the liver and the adipose tissue. And we obtained these biopsies from two groups, actually, which were we call healthy and unhealthy obesity, or obese persons, discriminated on the basis of currently known markers for insulin resistance. And when we analyzed these samples, it turned out that HO-1 is increased in the unhealthy obese subgroup. And this was in total contrast to what was expected from the literature and triggered all the subsequent studies that allowed us to pin down the mechanism.
So we used conditional knockout technology, which means that we can turn off the gene basically one tissue at a time in the tissue and time of our choosing. And what came out there was very interesting, and we had no idea. And one was that in muscle, in fat, in the beta cells, so the cells that secrete insulin, the molecules seemed to have no role in disease. But when we took it out in the liver, when we took it out in the macrophages, so the phagocytes that eat up and clean up our tissues and participate in the inflammatory process, suddenly we had huge effects on disease. When we had no HO-1, the animals were completely protected, even though through eating a high-fat diet, that's our main technique of inducing obesity in the animals, they had robust, full-blown obesity. They were heavy, they're fat, but the one group without HO-1 was cured from the disease.
So it's necessary. So if we take it out, we lose the phenotype. It's necessary first. And second, if we put it in the system, it's disease causing, so necessary and sufficient to cause metabolic disease. A very strong statement for such a molecule. HO-1's role in driving inflammation and insulin resistance is highly conserved between humans and mice. This is allowing us to use the mouse as a perfect model system because it fully recapitulates the human observations. And I think that this was one of the key success factors in this story. Inclusion of HO-1 in the diagnostic process of metabolic disease, type 2 diabetes, could enable us to screen a much wider population than it is possible now, because it's maybe simply to be diagnosed in a blood sample. So not by an oral glucose tolerance test that is much more complicated than a simple blood test. That this is important because nowadays more than 10%, even in Germany, of people with type 2 diabetes remain undetected.
HO-1 based therapies, so inhibitors, presumably inhibitors, would definitely be important for both stages, pre-diabetes as well as a mature disease form, because the inflammatory component seems to be responsible both for the establishment and then also the maintenance and the execution of the negative parts of the disease itself. And there's definitely many avenues of therapy right now, novel therapeutic strategies that are in clinical phases, which are almost exclusively focused on inhibiting the inflammatory process. And what we know here is that HO-1 is necessary and sufficient to drive it. So we know that targeting HO-1 alone, if it's successfully done, will prevent disease and treat disease. I mean, we can say that from the genetic evidence.
We know from data from several publications, mainly from pathologists, also on human samples, that HO-1 is indeed upregulated in the process of Alzheimer's disease, in Parkinson's, in the neurons, in the cells surrounding the neurons. So it is there. And people nowadays mostly think that it is kind of a self-defense of these cells, so that these cells try to defend themselves against cell death and ongoing disease processes. Based on our data, when we expand our data to these models, I would rather assume that HO-1 may be involved in the disease process, because we have clearly shown in evidence that HO-1 downregulates mitochondrial function, interferes with healthy cellular signaling processes. So yes, that would be very interesting to test if it also plays a causative role in these aging-associated diseases.
The next step for diagnosis is clearly I think the time frame is easy. That can be started right away because the molecular tools are there. We simply need more information, and that means from larger cohorts of patients, different types of diabetes, different types of insulin resistance. How generalizable is the principle? And then which diagnostics tools to use. From a therapeutic point, I think we're still a stage earlier. There are some major questions about how the molecule is affecting disease. How is it causing this disease process? We don't know if it's enzyme activity of HO-1, because it is an enzyme, or whether it's binding other molecules and acting as an adapter, or whether it's the fact that sometimes we find it in the nucleus, sometimes in the cytosol. So we need some more biological information to know what process do we really need to target with a compound or a drug therapy. And once that information happens, and I think that's a process of 1-2 years, so not a very long process, then the drug target design can happen. And there's some compounds out there already that can be tested ahead of time, but certainly for developing a treatment that would then be really targeted potentially at hundreds of millions of people, right? This would be something that's worth major investments in finding really the absolute best molecule.